Previously, we talked about the structure and the different types of collagen molecules. Now we're going to switch gears and talk about the way that our cells synthesize collagen and the way that our body breaks down collagen. So let's begin with synthesis. So in our body, we have specialized cells known as fibroblasts. And the entire point of these fibroblasts is to actually synthesize collagen. So we have different types of fibroblasts depending on what organ or tissue we're talking about. For example, in our bone, the fibroblasts are known as osteoblasts, and these generate type 1 collagen. While in our cartilage, for example, we have fibroblasts known as chondroblasts, and these generate type 2. Now, regardless of the fibroblasts that we're talking about, all of them actually carry out the same function in the same exact way. They generate the collagen. And so collagen synthesis begins inside the cell and ends outside the cell. And it's carried out in the following fashion. So let's begin in the nucleus. So this is the nucleus. We have our nuclear membrane, the nuclear pore. We have the cytoplasm. We have the rough endoplasmic reticulum, the Golgi apparatus, the cell membrane, and then outside the cell. So this is our fibroblasts. So within the nucleus, we have the nuclear machinery that undergoes the process of transcription. So we take the genes, transcribe the genes, and we form our mRNA molecule. The mRNA molecule leaves via these nuclear pores. It enters the cytosol of the cell, the fibroblasts, and then it associates with free ribosomes present in the cytosol. And this begins the process of translation. Now we begin the process of building that alpha molecule, that alpha chain. So remember, our single collagen monomer consists of 3 individual alpha chains. And so we begin building these alpha chains in the following way in the cytosol. Now, the initial molecule that we first begin producing is known as pre-pro alpha chain. The pre component of the pre-pro alpha chain means it contains this blue region on the N-terminal end of the translating polypeptide, and this is known as the signal sequence peptide. And this signal sequence peptide is used to actually direct this polypeptide to the rough endoplasmic reticulum. Now, when the entire free ribosome with this translating polypeptide and the mRNA ends up being transported to the rough endoplasmic reticulum because of this signal peptide, the signal sequence peptide is actually cleaved by an enzyme, and that forms the pro-alpha chain. So we remove the blue component, the signal peptide, and we form the pro-alpha chain. And we have many of these pro-alpha chains being formed at the same time within the lumen of the rough endoplasmic reticulum. Now, as we're forming that pro-alpha chain, we're also at the same time adding hydroxyl groups onto the proline and lysine to form the hydroxyproline and the hydroxylysine. And this is carried out by an enzyme known as hydroxylase. And hydroxylase uses a molecular oxygen for the oxidation process and vitamin C for the reduction process to actually attach those hydroxy groups. Now, in patients with scurvy, which is basically vitamin C deficiency, this process is affected, and people with scurvy cannot actually hydroxylate the collagen, and ultimately that affects the ability to actually form covalent cross-linkages at the end of this process. In fact, in patients with scurvy, they actually have collagen that is not very stabilized. And so, for example, those patients with scurvy actually have blood vessels that will bleed readily. And that's one of the signs and symptoms of patients with scurvy. So once we form the hydroxyl groups, some of those hydroxyl groups of the hydroxylysine actually are modified even further via the process of glycosylation. And this process involves adding things like glucose or galactose. So we add these sugar moieties, and ultimately they assist with the process of forming that final collagen structure. Now, Inside the lumen of the rough endoplasmic reticulum, many of these individual alpha chains are actually being formed at the same exact time. And once we hydroxylate the residues, 3 of these alpha chains, known as pro-alpha chains, can actually associate it to form a molecule known as procollagen. Now, a procollagen consists of 3 of these pro-alpha chains, and on the end of each of these chains, so we have the N-terminal end of the protein and the C-terminal end of the protein, these regions are known as propeptide regions, because these regions actually do not contain that helical structure. Instead, they contain disulfide bonds. So we have a bunch of disulfide bonds here and a bunch of disulfide bonds here. And these disulfide bonds in these propeptide sections, so this is known as propeptide, they're important to actually hold the structure of the procollagen together, at least initially. So once we form the procollagen that consists of 3 individual pro-alpha chains being held together at the ends by these disulfide bonds, they then go into the Golgi apparatus where we form the vesicles, and we essentially release the vesicles outside via the process of exocytosis. So once outside, we have these procollagen molecules that are acted on by enzymes known as peptidases. And what peptidases do is they snip the propeptides off. Because the entire point of these propeptides was to contain these disulfide bonds to hold the structure together. But now that they're outside, we want to remove them. And so the peptidase essentially clip them. So they clip this one off and they clip this one off, so that now we form that final collagen monomer. So now we have the collagen monomer without the propeptide ends, and they spontaneously arrange themselves to essentially form these stacking units. And this is known as a fibril. So we have one monomer here, another monomer here overlapping it, a third one, a fourth one. And this fibril isn't very stabilized. And so what we want to do finally is we want to form covalent bonds, strong bonds between the individual collagen monomers. And this is carried out by the enzyme known as lysyl oxidase. So lysyl oxidase essentially uses copper as a cofactor to form covalent bonds between the individual collagen monomers to ultimately form a very strong, reinforced, and stabilized structure. Now, in a disease known as Menkes disease, in which there's a problem in actually being able to take the copper and attach the copper onto this enzyme and other enzymes, the lysyl oxidase doesn't function as well, and so we can't form these cross linkages. And so the patients with Menkes disease are going to have problems associated with collagen instability. So this is the way we form collagen in our body. So to summarize, we have these specialized cells known as fibroblasts, which are actually responsible for initiating synthesis in a cell and finishing it outside the cell. So in a cell, we transcribe the genes to form the mRNA that ends up in the cytosol. We have free ribosomes that begin the process of translation, initially forming the pre-pro-alpha chain. And then once we cleave that signal sequence, we form the pro-alpha chain, which ends up being transferred to the rough endoplasmic reticulum, where the translation process continues. As we're continually translating that pro-alpha chain, we're also hydroxylating it and glycosylating the different residues, and eventually when we glycosylate and we hydroxylate, 3 individual proteins can combine to form a procollagen. The procollagen consists at both ends of these propeptide regions, in which we have disulfide bonds to actually hold the procollagen together. Then that transfers to the Golgi apparatus, and that forms vesicles, and the vesicles undergo exocytosis to actually end up outside the cell. Once outside the cell, we cleave the Propeptide ends with peptidases, thereby forming those individual collagen monomers that essentially spontaneously form these relatively stabilized structures known as fibrils, in which we have the monomers stacking like so. But to actually reinforce and stabilize the final structure, we have the enzyme known as lysyl oxidase that uses copper as a cofactor to actually cross-link and form those covalent bonds, thereby greatly stabilizing and reinforcing the structure. Now, the half-life of collagen is several months. And actually, collagen metabolism is actually a very dynamic process. So we're always breaking down and forming new collagen. And so the set of enzymes known as metalloproteases that are responsible for breaking down and degrading collagen are known as collagenases. So we have all this process that takes place inside and outside the cell to actually form collagen is then actually reversed by these collagenases that break down the collagen, and that makes way for us to actually be able to form new collagen. So this process of collagen remodeling is important, and we'll talk more about these collagenases when we'll talk about the process of forming granulation tissue. So in the next several lectures, we're going to talk about the different types of medical conditions that arise because of problems with collagen synthesis.